Hi students, in last class we discussed observing cells in onion, observing cells in matchstick. Who discovered cell first time? Robert Hooke in 1665. In today's lesson also we are going to discuss 2 or 3 more experiments. These are very important and easy. Observing human cheek cell. Observing human cheek cell. Observing human cheek cell. Observation of nucleus in cheek cell. Observation of nucleus in onion cell. So we are going to observe the cell in as well as animals and as well as plants. While we are doing an experimental activity, we have to follow some instructions. Like we already did so many experiments. Observing human cheek cell. Where cheek cell is present here? Here. This is a very sensitive part. Unless in the presence of teachers and doctor, we are not supposed to do this experiment. Before going to do this experiment, we should wash our mouth properly, because there are millions or thousands of bacteria present in our mouth. So we have to wash it properly with the water. Then first point, take toothpick or spatula or ice cream spoon. Any one of them. Toothpick. Take a toothpick, spatula or ice cream spoon. Through this toothpick, scrape little amount of inner lining of mouth. Taking a toothpick, small, like scraping, scrape the little amount of inner lining of mouth. Because it's very careful while we are doing this experiment, because it's very sensitive parts inside the mouth, you have to take a little amount. Then place it on slide. Place it on slide. Take the material, place it on slide. Then put a drop of water. Put a drop of water on the materials. Then put a drop of glycerin. Put a drop of glycerin. Then, before covering the cover slip, apply the blotting paper. Take a blotting paper. We should press it on properly on the material. Why we should press it? It will absorb the excess water present in the material. What happens when the excess water is present? Instead of this cheek cell, we will observe a large bubbles in the microscope. That's why it's very important to remove the excess water present in the materials. Then cover with cover slip. Observe under the microscope. Observe under the microscope. Simple activity, don't confuse. So, wash your mouth properly. Take a toothpick, spatula or ice cream spoon, scrape or take a little amount of inner lining of mouth with the help of toothpick, spatula or ice cream spoon. Place it on slide. Place it on slide. Put a drop of water, then put a drop of glycerin after a few minutes. Cover the cover slip Observe under the microscope. What precautions we should take while doing this experiment? Do not scrap material hardly from the inner mouth, because it will damage our inner mouth, inner linings. The blood will come away from this mouth. That's why we are not supposed to do our own. So we should remove the excess water present in the materials while applying the blotting paper, and we can apply tissue paper also to absorb the water present in the materials. Observe under the microscope. When we observe this, the presence of microscope without stain, we can see this diagram. So how many parts we can see in the cheek cell? There are a layer is there. This outer layer is called as cell membrane. Cell membrane. What is the another name of cell membrane? Also called as plasma membrane. Plasma membrane. The inner side, the middle of the cell, the dot-like structure is called as nucleus. Nucleus. Then the substances is present inside the cell is called as cytoplasma. Cytoplasma. So this is observation of cheek cell in human being. Observation of cheek cell in human being. Next we will discuss about observation of nucleus in onion. Observation of nucleus in onion. So before going to discuss that, this Felix Fontana, a discovery of significant observation of the nucleus. Including Felix Fontana, there are other scientists that did experiment on the observation of the nucleus. Finally, but they couldn't find out the functional or the structure of the nucleus. Finally, in 1831, Robert Brown discovered a prominent nucleus in the cell, in plant cell and animal cell. Robert Brown in 1831, he discovered nucleus. Nucleus is the largest organelles present in the cell. So it have a controller unit to control the whole cell. There are so many functions there about this nucleus. We will discuss in the next class. The next experiment, observation of nucleus in onion peel. Observation of nucleus in onion peel. Observation of nucleus in onion peel. This is also an experiment. Take onion bulb. Take onion bulbs. Onion. So you know the onion structures. There is some layers is present in onions. So you have to cut a small portion of onion. A small portion of onion. Cut it with the slices. Cut it with single slices. Then place it on. Take a small portion of onion. Take a small slices of onion. Place it on same. Place it on slide. Place it on slide. Cover with cover slip. Observe under the microscope. Also you can add glycerin. We should apply on Materials on blotting paper to observe the water present in the materials. So the same experiment we did in first experiment. Taking the material, place it on slide, put a drop of water, then put a drop of glycerin, apply with the blotting paper, it will absorb waters, then the materials will be spread properly on the slide, then cover with the cover slip, observe under the microscope. When we observe this in the presence of microscope, we will see that diagram, same diagrams. So we also have a membrane, plasma membrane. There is extra layer is present in onion, a plant that is called as cell wall. So nucleus is there, plasma membrane is there, and cytoplasm also is there. Cytoplasm. Simple activity. One more activity we discuss. Observation of nucleus in onion peel. Now we discuss observation of nucleus in chick cell. These are similar. They are almost similar. Observation of nucleus in chick cell. Now we already did the first experiment. Observe a human chick cell. How to observe human chick cell? Take a spatula, ice cream spoon, or toothpick. Scrub little amount of inner lining of mouth. Then place it on slide. Put a drop of water. Then put a drop of glycerin, cover with the cover slip, observe under the microscope. In first activity we discussed observation of chick cell, but in this activity we are going to observation of nucleus in chick cell, where the nucleus is present. Inside, in the middle of the cell, in animal cell, the dot-like structure inside the middle of the part is called as nucleus. Membrane is there, plasma membrane or cell membrane. The liquid substances is present inside the cell is called as cytoplasm. So these three activities simple. First one is observation of human chick cell, observation of nucleus in onion peel cell, observation of nucleus in chick cell. These are same activities, they have similar functions. So similar diagram, there is small differences between one diagram to another diagrams, but the most difference between the plant cell and the animal cell. In plant cell there is extra layer is there, that is called as cell wall. But in animal cell wall is absent. Cell wall is not there in animal cell. So this is about observation of nucleus in chick cell. Lastly, we will draw about diversity in cells. Diversity in cells. So we discussed about different types of cells like single cell, like amoeba, parameciums, chlamydomonas. Unicellular, uni is nothing but single. Multi is nothing but more than one cell is called as multicellars. So this both cells, unicellular also, they have a process like taking nutritions and excretions, sending waste materials, transportations, reproductions also takes place even it is a single cell. So this all like process will take place in this singular cells. Even growth also will take place. Amoeba is a single cell. But multicellars, they have more than cells. Like plants and animals are multicellular organisms. They have systems in their body. The transport system is there, excretory systems is there, respiratory systems is there. They have different systems in their body. They are given in diverse cells. Diagram of Chlamydomonas. Diagram of Chlamydomonas. And diagram of amoeba. Amoeba doesn't have any shape. It's called as irregular shape. They have a false feet. It's called as pseudopodia. Nucleus is present. Contracted vacuoles. I have contracted vacuoles. I have flagella, nucleus, cytoplasm is there. These are two diagrams is important. One more diagram, that is amoeba, paramecium. So the paramecium diagram is not there in your book, just write. So there's three diagrams. Chlamydomonas. Amoeba. Paramecium. Explanation not given in any class. There in further classes. The diagram is important. Draw this three diagram and the activity. Observation of sick cell. The second activities both are same. Just write one activity and you should compulsory draw this three diagram. Chlamydomonas, amoeba and a paramecium. Thank you, student. So this is the today homework.
Stop pregnancy. And close the outside.